The cell is the smallest unit of life, so we're going to look at the cell and how it divides. There are three major parts of a cell. We have the cell membrane, we have the nucleus, and the cytoplasm. The plasma membrane, or the cell membrane, maintains the boundary. You'll remember that life forms have to be able to maintain a boundary. It also helps transport materials in and out of the cell. The nucleus is the control center. The DNA is housed here, and that controls all of the activities of the cell. And the cytoplasm is the material between the nucleus and the plasma or cell membrane. In the cytoplasm, we see the organelles. These are the structures where all of the chemical reactions occur, and also the cytoplasm may have inclusions. These are materials that are stored by the cell for later use. The cell cycle is basically the life cycle of the cell. Most of the time, the cell is in interphase, and interphase is divided into three components, G1, S, and G2. Mitosis is a small piece of the cell cycle. Mitosis is when nuclear division occurs. And right at the end of mitosis, we have the division of the cytoplasm. This is known as cytokinesis. Interphase, where the cell spends most of its time, has as the beginning the G1 phase. This is the phase of cell growth and metabolism. This is basically the cell being the cell. This is the variable stage of the cell cycle. Cells may spend a little bit of time here, or they may spend a lot of time here. Some cells lose the ability to divide, like nerve cells and cardiac muscle cells. These cells are permanently stuck in G1 phase. If the S phase starts, that's because the cell has gotten the signal that it's going to divide. Because during the S phase, which stands for synthesis, the cell makes a copy of all of the DNA. If the cell is going to divide, each new cell has to have an exact copy of the DNA. Then the second growth phase, the G2 phase, is when the cell goes through some additional growth. If the cell is going to divide, it needs lots of cytoplasm, and it also replicates the organelles at this stage so that there are enough organelles to go into two cells. During the S phase, DNA replicates. The DNA makes a copy of itself, or the chromosomes replicate. We start out with one chromosome, and through a process of DNA replication, we end up with two chromosomes. Now, the exact copies are held together by a structure called a centromere. And while they are held together by the centromere, we refer to them as chromatids. In order to see mitosis, we need a group of actively dividing cells, and we use something called a whitefish blastula. This is the stage where all of the cells are actively dividing. And if we cut this in cross-section, we can catch cells in various stages of cell division. In interphase, which is the period of cell growth and metabolic activity, we can see the cell membrane, the cytoplasm, and the nucleus. We clearly see the nuclear membrane, and we don't see too much going on in the nucleus. It's pretty uniform in structure. As we begin mitosis, the first stage is prophase, and one of the things that happens is the chromatin begins to condense. That is, the chromosomes start to become visible, and that's this darkening you see in the nucleus. Also, the nuclear membrane begins to disappear, and if you'll notice in this cell, we don't quite have that sharp boundary of the nucleus any longer. The centrioles begin to develop. We begin to see them, and they start to develop spindle fibers. During the next phase of mitosis, metaphase, we see the centrioles. They have moved to the opposite ends of the cell, and they have developed spindle fibers. The chromosomes are all lined up here at the equator. The spindle fibers attach to each chromosome, as well as attaching to the cell membrane back here, so that the centrioles are anchored in the cell. During the next phase of mitosis, anaphase, the centromeres divide, so those chromatids are no longer held together, and the individual chromosomes can be pulled apart. The spindle fibers begin to shorten, pulling the chromosomes to opposite ends of the cell. And then during telophase, we begin cytokinesis, that little in-pocketing of the cell membrane right here. And here it's pretty much almost complete. Notice that we have the nuclear material condensed back up. The nucleus is going to reform. The centrioles and the spindle fibers will disappear, and the chromosomes will condense back into chromatin. We'll start to see that nucleus go back to not being so prominent anymore. Mitosis gives you two identical daughter cells. The two cells are identical to each other, and they're identical to the original mother cell. Mitosis is used in the body for growth and repair. This is how we replace cells with exactly the same kind of cell.